I'm really excited to be part of this webinar series again and to have the opportunity this time to present some of my research on diatoms from the East African Great Lakes, one of the most remarkable biodiversity hotspots and home to some of the most iconic adaptive radiations on our planet. So, from the earliest stages of the planet formation and its differentiation to the birth of where life first emerged, cyanobacteria soon develop oxygenic photosynthesis. This revolutionary process transformed the face of our planet by enriching the atmosphere with oxygen and ultimately setting the stage for evolution of complex life forms and major bursts of biodiversity, such as the Cambrian explosion, the colonization of land, and the Mesozoic marine revolution, during which time the tiny yet powerful glass wall powerhouses of diatoms first emerged and began to take over a major share of this essential function. Today, diatoms produce about 20% of the oxygen we breathe and help sustain nearly all aquatic ecosystems through nutrient cycling and climate regulation. A key to this success lay in the ability of diatoms to build intricate silica shell, tiny glass-like houses, where key morphological features such as areola and spines that enhance protection, buoyancy, and nutrient exchange are expressed. But their true advantage resides within the cell itself, where chloroplast, mitochondria, and lipids drive highly efficient physiological processes such as photosynthesis and long-term energy storage. Several of these traits have been considered evolutionary innovations that may have likely enabled diatoms to rapidly spread and adapt to nearly every aquatic environment on the planet, becoming vital to the ecosystem functioning and diversifying into an estimated 200,000 species today. This remarkable diversification can in part be tracked to the very nature of diatoms themselves, a unique blend of microbial simplicity and eukaryotic complexity. On one side, their evolution has largely been shaped by slow, gradual processes such as horizontal gene transfer and endosymbiosis, but on the other hand, their eukaryotic complexity and ecological versatility may have provided the foundations for bursts of diversification, including episodes of adaptive radiation. And yet, despite this potential, adaptive radiation has never been clearly demonstrated in diatoms. This is largely because research has traditionally focused on their microbial nature, and partially because we still know surprisingly very little about their general biology, evolution, phylogeny, and genomic architecture. We still don't know which of these traits may have acted as key evolutionary innovations and how these traits enable diatoms to exploit such a wide range of ecological opportunities and niches, which are conditions that often triggered adaptive radiations in other lineages but have never been clearly linked to diatom evolution. This is especially striking given that adaptive radiation is widely recognized as one of the most powerful evolutionary processes driving biodiversity. It occurs when a lineage rapidly diversifies to occupy a range of ecological niches, often triggered by the colonization of a new habitat, the evolution of key innovation, or the extinction of competitors or predators. These dynamics are most clearly observed in isolated ecosystems such as islands and ancient lakes, where geographical isolations, small ancestral populations persisting over millions of years, and relative environmental stability create ideal conditions for tracking repeated evolutionary processes. Among the ancient lakes, those in the East African Rift Valley, especially Lake Tanganyika and Lake Malawi, stand out as home to some of the most iconic examples of adaptive radiations. While the spectacular radiations of cichlid fishes in this lake are among the best known and perhaps the most extensive ones in the aquatic realm, These systems have also likely fostered radiations in other less explored groups such as diatoms, given their primarily role in supporting the food web on which cichlids and many other organisms heavily rely. So building on this idea, during my previous postdoctoral project at the Senckenberg Natural History Museum in Frankfurt, I began to explore the diatom diversity in these unique biodiversity hotspot regions with a goal to investigate their diversification trajectories and to see whether any of the targeted groups show signs of adaptive radiation. To do this, I collected a wide variety of modern samples at different depths along the shores of Lake Tanganyika and Lake Malawi during several field expeditions and performed DNA isolations from individual cells of the freshly collected samples directly on site using Calyx 100 solution. But to gain a better understanding on the endemism and the uniqueness of the lake-specific species, I also collected samples from the surrounding freshwater habitats as well as marine habitats along the Tanzanian coast of the Indian Ocean. But at the same time, to support this assessment and to identify potential extinct species, I analyzed local outcrops from the Chivando beds of Pleistocene age in northern Malawi in a very close proximity from Lake Malawi, as well as drill cores that has been retrieved in previous years by other researchers from both Lake Malawi and Lake Tanganyika. The sediment core from Lake Tanganyika is relatively short. It's only 8 meters long that has been retrieved from about 300 meters water depth at the Kalia Horst region. It spans only the last 60,000 years, which is relatively little in evolutionary terms if we consider the estimated age of Lake Tanganyika between 10 and 12 million years. But nevertheless, it provided us with a valuable information on the preservation potential, offering indirect insights into the timing of emergence of the species as we will estimate from the molecular clock analysis. In contrast, the Lake Malawi core has been retrieved from a little bit greater depths of nearly 400 meters and spans on the last 1.2 million years, which is close to the formation of the estimated age of Lake Malawi, which is between 2 and 5 million years. This allows us to track evolutionary changes in real time and to independently infer speciation and extinction time using bird-dead preservation model, which uses presence-absence data and fossil occurrence frequency to estimate uncertainties in preservation for each species. So I focus on the genera Diploneis, Afrocymbella, Capartogramma, and Sellaphora to capture lineages with contrasting lifestyles and uneven species richness with the goal to identify similarities and differences in their evolutionary trajectory and explore the factors shaping them. So as a first step, we conducted a comprehensive taxonomic assessment for each genus, starting by exploring the existing literature and supplementing it with our own morphological observation and morphometric analysis and genetic data from the two most widely used genetic markers for diatoms, RBCL and 18S. This integrative approach will enable us more accurate species identification and reveal previously unrecognized and overlooked diversity. This phase of the work was unexpectedly time-consuming and particularly challenging, especially within the genera Sellaphora and Diploneis. But to better understand the number of lineages and their evolutionary relationships, we reconstructed a molecular phylogeny using these two genetic markers, 18S and RBCL, from 360 rapid diatom taxa, combining GenBank records with our own field-collected samples. This analysis revealed many closely related East African Diploneis species with small genetic distances, forming a well-supported monophyletic group comprising two distinct and well-supported clades, one including species from both Lake Tanganyika and Lake Malawi, and the other clade including species from the surrounding areas of the lake. The number of lineages and their phylogenetic relationships were supported by our morphological observations using both light and scanning electron microscope, as well as morphometric analysis based on landmark and pseudo-landmark data from 30 individuals per taxon. The B plot of the first major axes of morphological variance shows that each lineage occupies a distinct morphospace, mainly reflecting differences in valve outline and striate density. But beyond these differences, ACM revealed additional diagnostic features, in particular the presence of fin-like ornamentation on the exterior wall of their glass shells. This striking feature is found exclusively in the lake species, and it's absent in the species from the surrounding habitats and elsewhere globally, except for very few species from Lake Baikal. However, we believe that these Baikal species may have acquired this trait independently through convergent evolution, as they are likely descendant from distinct ancestral lineages. We don't know why they developed this feature, but we assume that it could be functionally driven and related to more efficient light absorption in the deeper, low-light benthic habitats of Lake Tanganyika, in contrast to the surrounding waters, and or to serve to optimize adhesion on the surface in face of predation or strong water currents in the lake, maybe in a manner somewhat similar to that in Lake Baikal. So based on this combined evidence, we describe all these taxa as new to science. So we described 21 species from the modern records of Lake Tanganyika, the species in pinkish color, 4 species from the modern records from Lake Malawi, and 2 presumably extinct species from the fossil sediments of Lake Malawi, the species in yellowish, and 3 species from the surrounding habitats, the species in blue. Then we screen the fossil records to determine the earliest appearance of Diploneis species. In Lake Tanganyika, although, as I mentioned, it is relatively shallow in evolutionary terms, still we were able to recover more than half of the modern species, plus 9 seemingly extinct species. Given the limited temporal depth of the core, the bird death preservation model captured only a little variation in speciation and extinction times. But in contrast, in Lake Malawi, where we overall observed a lower number of Diploneis species, all of them extant, plus 11 seemingly extinct species, the model here performed much better because it captured a clear variation in speciation and extinction times, but with low preservation uncertainties, which well align with the first and the last occurrence of the species in the course. We did not find any of these species from the course or from the modern records in the Chibanda outcrops in northern Malawi, close to the lake. We then incorporated all these newly described extant Diploneis species into a time-calibrated molecular phylogeny based on 11 genetic markers from 316 diatom taxa, adding the extinct species a posteriori based on their morphology and stratigraphy. So we defined branch lengths using the first and the last occurrence in the fossil record as a minimum and maximum age. And we found that the monophyletic Lake Diploneis group, including lineages from Lake Tanganyika and few nested within it from Lake Malawi, to exhibit short branch lengths, suggesting a recent and potential adaptive radiation. This roughly coincides with the formation of Lake Tanganyika, as the cratable intervals lay between 31.3 and 40.6 million years. The scenario of adaptive radiation is further supported by this fin-like structure, which may represent a key evolutionary innovation driving diversification within the benthic habitats rich in ecological opportunities and niches where these species thrive. The potential role of these Fin-like structures as key evolutionary innovation is supported by the variation in size, number, and shape across the species, as well as by their significance in factor analysis of mixed data using 27 continuous and categorical traits. However, to assess their functional and evolutionary role, we plan phenotype environment correlation analysis and diversification rate estimates. So then using the same phylogenetic framework based on the data set of two genes, the RBCL and 18S from the 316 rapid diatom species, we also analyze sequences from nearly all Afrocymbella species collected from the lakes. So our molecular data reveal closely related but distinct species with small genetic distances, forming a well-supported monophyletic group, significantly less species-rich than Diploneis. This number of lineages were also supported by our morphological observations, both by LM and ACM, as well as morphometric analysis, again based on landmark and pseudo-landmark data on 30 individuals per taxon. The morphospace B plot shows that each lineage occupies a unique position, primarily reflecting differences in the shell symmetry and tumescence. So based on this evidence, we describe 5 new Afrocymbella species, 3 from the modern records and 2 from the fossil records of Lake Malawi, in addition to the already known ones for the rift. We found a larger number of Afrocymbella species in the Lake Tanganyika core, likely because the core has been retrieved from the shallower areas and closer to the rocky substrates where these species thrive. But in contrast to Lake Malawi, where we found 2 presumably extinct species that apparently disappeared with the onset of the last mega drought, when the water volume of Lake Malawi decreased for at least 95%. In Lake Tanganyika, we did not detect any extinct species over the past 60,000 years. However, further analysis may reveal them, especially if the planned deep drilling project on Lake Tanganyika go ahead. This project aims to recover sediments spanning more than 5 million years, which will allow us to track evolutionary changes over time and to potentially identify or detect extinct species. So we then incorporated all these extant and extinct Afrocymbella species in the same time-calibrated dataset compressing 11 genes and 316 diatom species to reconstruct their evolutionary history using an approach similar to that we employed for Diploneis. The resulting monophyletic Afrocymbella clade exhibits short branch lengths suggesting a recent and possibly rapid diversification that may have occurred between 17.3 and 9.45 million years, which again roughly coincides with the formation of Lake Tanganyika. But to investigate whether this pattern reflects adaptive radiation, we plan to estimate diversification rates for Afrocymbella and its closest relative, which appear to be Gonphonema species from the rifts, specifically from the Gonphonema africanum groups, which include Gonphonema padocki, kelhami, and africanum, which may represent a distinct genus or appear to represent a distinct genus, but this really requires further analysis. So, to further support the scenario of adaptive radiation and identify potential key evolutionary innovations within the genus Afrocymbella, we scored 29 continuous and categorical traits. Using again factor analysis of mixed data, we found that the valve symmetry and tumescence, which were already highlighted in the landmark analysis earlier, are promising traits. However, similarly is for Diploneis to verify their adaptive significance and volume, we will very soon correlate them with various environmental variables. So next we turn to a much smaller yet fascinating genus with the high species richness here in the Rift Valley, Capartogramma. Globally, only 7 species have been recorded so far. Most of them with very restricted distribution, except for Capartogramma crucicola. In Lake Tanganyika alone, 3 species coexist: Capartogramma rhombicum, casteni, and ampheroides. Our goal was to reconstruct their evolutionary history and relationships, and to clarify their position within the broader diatom phylogeny. Identifying their sister genus would also allow us to compare speciation rates and explore differences in evolutionary dynamics, although we remain cautious about the statistical power of such comparisons. But using the standard 2 gene markers, we constructed a molecular phylogeny, including representative genera, and the results revealed small genetic distances among the 3 Capartogramma species, clearly supporting their phylogenetic relationships. Interestingly, the 2 endemic species from Lake Tanganyika, Capartogramma ampheroides and rhombicum, are more closely related to each other than to Capartogramma carsteni, which does not correspond to earlier assumptions based on morphology. We identified Diploneis as the sister genus of Capartogramma, but with only one sequence, it is difficult or even impossible to estimate diversification of the genus, and maybe perhaps of both genera. However, to explore the timing, we calibrated these trees using the same fossil dataset by Nakov et al. 2018, and we found that Capartogramma carsteni diverged around 26 million years ago, likely outside the lake, while the 2 endemic species split about 6.5 million years, pointing to a colonization of the lake followed by in situ diversification. However, broader sampling will be needed to confirm any of these scenarios. Sorry. So looking into the fossil record, we see hints that speciation may have occurred within the lake itself. All 3 Capartogramma species appear consistently over the last 16,000 years. While this time frame doesn't allow us to pinpoint exactly when and where they originated, it does provide an important context. The relatively low preservation bias and the consistently high abundance of the species suggest that they may have established themselves in the system earlier, which potentially aligns with the divergent times derived from our molecular data. And although it's not conclusive, these patterns are somewhat consistent with a scenario of in situ diversification, but whether this diversification was adaptive, however, remains unclear, particularly given the low species richness of the genus. A more robust test of this hypothesis would require the broader taxonomic sampling, older fossil records, and integration of ecological and preferably genomic data. Then we started exploring the species richness within the genus Sellaphora together with a bachelor student from Macedonia. And using light microscopy, we uncovered surprisingly high and previously overlooked diversity, identifying more than 14 distinct morphotypes from Lake Tanganyika alone. However, more detailed analysis, including morphometrics and genetic data, as we conducted for the other genera, will be needed to confirm their taxonomic status more accurately. But although we have begun gathering more detailed observations using scanning electron microscope, there is still much work ahead to fully resolve the taxonomy of Sellaphora. It's like exceptionally high morphological diversity makes it as a really challenging but exciting task. So, however, we have been able to identify some of these modern Sellaphora morphotypes in the fossil records from both Lake Tanganyika and Lake Malawi. So far, we have not observed any extinct species. However, further analysis is likely to reveal them, especially if, as I mentioned earlier, the planned deep drilling in Lake Tanganyika does indeed take place. So While these species of Sellaphora and the other genera likely involved in situ within the rift and most probably originating from the ancient cradle of Lake Tanganyika before colonizing and diversifying elsewhere, Sellaphora and Diploneis stand out as the most promising candidates for having undergone adaptive radiation. This is particularly evident for Diploneis, where in addition to the apparent monophyly of the species from Lake Tanganyika, their endemism, their rapid diversification, we identify a potential key morphological innovation, the emergence of fin-like ornamentation that may have driven diversification within the lake. Although we have made a progress in uncovering this process in Diploneis, the limited resolution of our current multi-locus phylogeny really underscores the need for higher resolution genomic, functional, and ecological data to test this hypothesis more rigorously. So to address these key research gaps regarding Diploneis and microbial adaptive radiation more broadly, I started the Swiss National Science Foundation Ambizione project 6 months ago, which will run for the next 4 years at the University of Basel. And this project aims to create an integrative framework that will combine morphology, physiology, genomics, transcriptomics, and computational biology within the diatom genus Diploneis to achieve 3 key objectives. Evaluate the adaptive nature of the Diploneis radiation, identify key evolutionary innovations and their underlying genes, and determine the function of key evolutionary innovations. So I will be working on this project together with a student from the Masaryk University in Brno, who has a background in bioinformatics. She will be joining our group to start her PhD on this project. So I will briefly outline the general idea of the project. So we will return on the field to sample unknown Diploneis species, both in cultures as a single-cell DNA isolates, along with the sister lineages from the surrounding habitats. These samples will be used to isolate, amplify, sequence, and assemble whole genomes using single-cell whole genome sequencing. And using this data, we will construct a genome-wide, time-calibrated molecular phylogeny that will incorporate the previously identified extinct species in order to confirm the monophyly of the Lake Diploneis clade and assess its diversification rate in comparison to its non-radiating relatives. And finally, we will investigate whether specific traits functioned as key innovation that facilitated this radiation by evaluating their adaptive value through phenotype environment correlation analysis. So we will consider several traits, but our primarily focus will be on the fin structures that are unique for the lake species, along with the physiological traits such as sterol composition, which are membrane-bound structural lipids that are essential for the membrane functioning in this group. So, but to further assess whether these candidate traits functioned as key innovation, we will also investigate the genetic changes underlying their emergence and adaptive significance using comparative genomic framework that will include the identification of genomic differences such as SNPs, gene gain losses, structural variants between the radiating and non-radiating sister lineages. Then we will look for signatures of positive selection in the coding region across the radiating clade and intend to link genomic variations to traits and environmental conditions using phylogenetically informed association tests and selection scalings. This will help us to pinpoint SNPs and genes such as those related to fin morphology and sterol composition that show adaptive relevance within the lake system. And in the final step, we will try to determine the functional role of candidate traits and their associated gene networks through experimental assays and gene expression analysis using both single-cell and bulk RNA sequencing to capture individual and population-level responses in 4 Diploneis species. So 3 species from Lake Tanganyika with varying degree of the fin structures and 1 control without fins from the nearby habitats. So by doing this, we believe that we can slowly start to uncover the functional roles and genetic foundations of evolutionary innovations and traits more broadly. This will help us to understand how such traits contribute to diversification and drive adaptive radiation in the larger story of eukaryotic complexity, and also will try to help us why some traits spark bursts of diversification and others not, uncovering the mechanism underlying the uneven pattern of species richness in these tiny but yet powerful organisms that play a vital role in climate regulation and ecosystem functioning. And with that, I would like to thank everyone who has supported this work, both directly and behind the scene. It has been truly a collaborative effort to achieve these results so far. And I'm especially grateful to the funding agency whose support has made this research and the many sampling expeditions over the past 7 years possible. And although not always in the spotlight, I would like to thank the many wonderful people, some of whom are unfortunately no longer with us, whose kindness, generosity, and support made this work possible on ground in the beautiful lands of Africa, where I'm very happy to be returning, hopefully already this summer. And of course, I would like to thank you all for your attention and for being here today.
Well, thank you so much, Etsy. That's wonderful. What an amazing project and what an amazing landscape to be able to work in. That's just great. If folks have questions, feel free to use the raise your hand feature, which is down in the bottom in the react button on the bottom of your screen, or you can type questions into the chat as well, and we'll pose them to Etsy and see what she has to say. I have a quick question. I long ago I worked on Lake Baikal and looked at those long sediment records, and there were so many depths where you wouldn't ever see a benthic diatom. Do you see the same thing in these African lakes where it is just thousands and thousands and thousands of planktonic species and then one benthic? Or how do you find these benthic diatoms in sediment cores from Lake Tanganyika in Malawi?
Mm, Well, in Lake Malawi, we find fewer number of benthic species in comparisons to Lake Tanganyika, simply because I think that the core in Lake Tanganyika has been retrieved from the shallower parts. Still, 100
Mhm.
meters make a difference. Surprisingly, in Tanganyika, we find quite a bit of the of benthic species. So I don't know. I will not say it by heart, but I mean, being just able to recover more than half of the modern species in the fossil sediments of Lake Tanganyika already says something. So yeah, it's surprising. Like I think maybe because of the water currents,
Hmm.
uh, they kind of are able to reach the drilling location much easier. Plus, in the area where they drill the lake, it's getting really deep, so it's
Hmm.
not so far from the littoral areas of the lake.
Okay.
In Malawi, it's a bit different because in Malawi, it's more on a distance from the littoral, from the shores. And there we find fewer benthic species, but they are still, I mean, it's not as rare as apparently in Lake Baikal. So I guess it really depends on the drilling location,
Hmm.
on, yeah, on the drilling location and yeah, and probably on the redistribution patterns, I guess. I don't know, Mark, you will know this better because you are
No, no,
working
I
more on this field that I can just say that, yeah, we find them, no, we don't find them. And like with my really limited knowledge in geology, I can just imagine how this redistribution could work out. But really, I think that you will know this better than me.
No, it's interesting that you're able to get such good diversity of the benthic stuff. It was so different in bical. You'd see one benthic diatom, you go, oh, that's a cool diatom. You'll never see it again, ever.
Really? Really?
So
But from where, from which depth do
we
you?
were we were I don't I we were we were on the hump between the two basins, and it was, but they were, I don't know how deep it is. They're 700 meters or something. It was deep stuff.
Okay,
So, but
okay, okay.
Matt, go ahead and turn on your mic and pose your question if you would. And turn on your camera too. Let's see you, Matt.
Yeah, that would be great that we
Sorry.
see you.
There he is.
Hello, Matt.
Hey, Matt.
So Etsy, absolutely fascinating talk again, as usual. I'm going to try to just pick one thing to talk about here, so I don't dominate this session. But the fins.
Yes.
So you had speculated that the fins in Diploneis had potentially something to do with depth. Depth, you said?
Yes. So, well, yes. So our two assumptions were that it could be related with the more efficient light absorption.
Okay.
Second one, to for a better adhesion or attachments on the sediments because of strong water currents. Uh, the water currents, I don't know. You have been to Lake Baikal, like maybe some of you have been there, but I have only seen it from videos and images where the water currents are similarly strong as in Lake Tanganyika, and that's why our assumption was that maybe it could be related to that, but we don't know. We don't know. So that's why one of the reasons of the second package or objectives from my ongoing project now is to try to really determine the functional role. Why are they doing that? Why do they develop this feature, and what kind of function they could have or serve to this group? Yeah.
Right. So we've got a potential analog in the epizoic diatoms. So the
Really?
ignanthes we see on some of these epizoics, we do see fins, which I'm not familiar with seeing on some of the non-epizoic ignanthes species. So in particular, Pseudogroenlandica, which was
Mhm.
described, I think, from the Mediterranean without any sort of fins whatsoever. But I mean, we've pulled it from a couple of different animals where everything else about the frustule says, well, this is Pseudogroenlandica, but it has a very distinct set of fins on the rafeless valve.
Mm.
So yeah, I don't know what that means yet, but it was interesting to hear you bring up the fins as a potential innovation there, and just interesting to hear another group of raphid diatoms where these things exist. These finned Diploneis, have you gotten them in culture yet?
Sorry?
The finned Diploneis, have you gotten them in culture yet?
Yes, so I tried to culture few species from Lake Tanganyika under extremely poor conditions. But
Sure.
surprisingly, they're still thriving. They are not very healthy. They don't look very good, but they're still thriving after 2 years. So that's why I think that if we optimize these conditions, we can obtain a reasonable growth that we could do some of our plant analysis with them. Not only for the genomes, but also to try to do the experiments. And most importantly, which I'm a bit skeptical, but let's hope that it will turn out possible to try to quantify the sterile composition within them, because then you need a lot of like amount of culture for that to measure that the composition and the distribution in this trait.
Yeah, maybe not as much as you think, but that's something we can talk about later. But the cultures have the same sort of fin structure that they do in the wild.
Sorry, sorry, I don't know why Matt, I
The
don't
the
hear you very
cultures,
well. Yeah.
I'm talking very fast. I'm sorry. I will slow
No,
down.
somehow it's breaking. I don't
The
know whether it's me or yeah.
yeah, all right. The cultured Diploneis, they still have fins. Same fins.
I have not observed them under the scanning electron microscope, not yet. It was really just,
Okay.
for me, that moment was just a pilot to see whether they will survive or not, because we have been trying, as you know, with Elizabeth to culture some species from Lake Ohrid, and it was always an extreme challenge and failure. So I have to admit that I was extremely surprised that they are thriving, and especially inside. I mean, I'm keeping them in an oven in the basement with a really poorly controlled conditions, and they're still alive. So this is, for me, it's like really surprising thing. But I will go back on the feces, just one more thing. So but we see, like, you observe that in different groups, but what we see in Achnanthus or Achnanthidium, or I don't know, maybe I'm wrong with the group that is living in both Lake Tanganyika and Lake Malawi, there are longitudinal stripes that look very like the features are different, but to me it appeared that they have a very similar function. So they're grebens with these finces that maybe really helps them like to sustain themselves in the water column or attached to the surfaces. But this is, I don't know. There are so many other like things for the African lakes. I don't know. Some of you maybe have seen, maybe not. Like all of them have asymmetric, slightly asymmetric shape. And I don't know what drives this asymmetry. Either is the currents or something else. I don't know. I don't know whether in Baikal they have seen this trait, but basically in Diploneis and with other groups, we see a slight asymmetry in the valves, basically in all species.
Even
Thank
in
you. Again, like, well,
no, I was gonna say even in
just like I said, I
Diploneus,
don't.
you get
Yes,
asymmetry in
yes,
Diploneus?
yes,
Oh wow.
slightly.
Go
Yes,
ahead, Matt.
slightly. Slightly, Mark,
Oh,
yes. So this way it's like the other thing for me is like the origin of Afrocymbella. It's like a gomphonemoid, apparently a gomphonemoid lineage.
Mhm.
They developed the asymmetry, like the apical asymmetry, apical or how? So the asymmetry. So and it's kind of a question why? Why to me? Why develop that feature? How that formed for what reason it formed. So this would be one of the project, and a student from Kartik's group is coming now that in June through Luke Hector's grants that they found one weird gomphonemoid genus in India, and then we want to place it in the Cymbellales to see how related or whether it's related to Afrocymbella, and try to basically in future reconstruct some of this trait evolution, the mode and the tempo of trait evolution within the group.
just one quick last thing about the Capartogramma molecular phylogeny. If you want more star Aeneid sequences, shoot me an email. I've got a lot of stuff that isn't published yet. So
Yeah, that will be great. I mean, we just submitted with Jeff, a student, PhD student, a manuscript where we were kind of talking about the evolutionary history and the phylogenetic systematic position of Capartogramma. So we submitted that one. But I have another plan, which I want to reconstruct the evolutionary history of the family Stauroneidae, and I think this will be great. So maybe we can talk about that, because it will be great if we can add more data on that. Yeah, this will be amazing. Thank you,
all right.
Matt. Thank you,
Thank
Matt.
Good. Feel free to raise your hand if you have any questions or type it in the chat.
I'll go for it,
Hey,
Mark.
Sarah.
Go for it.
Yeah.
Oh, I didn't see your hand up
Hey,
there, Sarah.
Sarah.
Sorry. Go
Good.
for it, Sarah.
Etsy, thanks for a fabulous talk. It's so interesting, and like Mark said, I could monopolize the questions here. With adaptive radiation, that's really tricky. I mean, how do you rule out that these fin-like structures and their variation originated, say, from genetic drift, from isolation of these basin. I don't know actually what the geologic history of the lakes is, but I imagine sort of sub-basins could have some isolation in old lakes. It seems really tricky to eliminate those other things, and you focused on function. How do you think you'll show function of these fin-like structures, and how they would, say, function differently either in time or space as differentiated,
Yeah.
to give some advantage in one situation versus another.
So, difficult question, very complex question, many aspects within it. So first, theoretically, one of the aspects for showing whether something is a key innovation is that you have to show that there is a positive correlation of the traits with a certain environmental variables. Basically, that environmental variables were selected for the trait to develop in order the species to fit better in a certain environmental conditions or niche. So we will do that by phenotype environment correlation analysis, meaning that we will try to gather as much as possible of the environmental data and try to correlate them. So what we will use for the lake, so I will not gather environmental data, because just gathering the environmental data by itself, it's a completely different project. It's a project for itself. So we will use the already published data, which includes light spectrum, pH, phosphorus, nitrogen, silica, all these variables. And then we will correlate them with the traits to see whether we'll find a positive correlations. Once we find a positive correlations with the genetic data, we can see which are the genes that are responsible for that trait and see when it evolved, because this is the most important and key for key innovation. When did it evolve? Whether it evolved before the actual diversification, during the diversification, or it was already present as a trait in the ancestors, which then immediately tells you that is not key evolutionary innovation.
And how do you get it? How do you get the time aspect of when it evolved?
So, well, we time-calibrate phylogeny. So we will construct a time-calibrated phylogeny.
Okay.
So then we'll include all the genomes, then we'll time-calibrate the phylogeny, and then we can see when did they evolve. Then we'll put all these traits on the phylogenetic tree, and we can see when the trait emerged within the phylogenetic tree. And then once we know which are the potential environmental variables that were kind of basically redefining the potential emergence of these traits, we will expose them in experiments to see if we alter the environmental conditions, how these finces will respond. So we will use the transcriptomic profiling for that in order to identify the functional role of these finces. And plus also sterols, because sterols are commonly known that they probably evolve through horizontal gene transfer, because they use them mostly for adaptation in adverse environmental conditions. So
what's the function? What's the function of the sterols?
Like they are bounded in the membrane, so they help them, like mostly for structural things, like that they kind of better fit or better control nutrients exchange and every basically function within the membrane. And that's why they assume that they could most likely be derivated through or gained through horizontal gene transfer and not really from key innovation. But that's what we want to test, whether it's indeed like that or not.
Yeah.
But next time when we meet, I explain you step by step, each step that we will do, and then try. Maybe it's like all these packages. Look, Sarah, like many of these things are also new for me. I have never done genomics. I have never done transcriptomics. So that's why I came here, so that I can learn all these aspects. So many of these packages, also work packages, are very novel, very new to me. But I'm very excited to be learning
That's
that. I
excellent.
mean, I learned quite a bit geology, so I guess that this should also be feasible.
That's awesome. Thanks,
Yeah.
Etsy.
Mm, I love it. Hey, Etsy, I was unclear when you were sampling the other habitats around around Eastern Africa there. Were you
Yeah.
seeing endemics in those areas as well, or were those more and I guess my other question is, are there other like cosmopolitan Diploneis that are there? You know, the generic ovalis is hanging around and puellalis
Mhm.
and stuff like that.
I have not seen any of the commonly widespread Diploneis species in these sampling
Huh.
locations. So most of them are kind of exclusive, but how exclusive they are is really difficult to say, Mark. As you know, Africa has been very poorly studied, so there is a lot of gaps in knowledge. So in order to say whether something is exclusive or regionally characteristic, I mean, I was doing as much as possible of detailed survey in the area to try to understand at least whether the species that we find in the lake are endemic or unique to the lake than what is outside. But what is outside, whether is regionally constrained there or, I mean, geographically constrained, this is very difficult to say. I think it's almost impossible at the moment to say. But with the data that we have, yes, it's very unique.
Neat. All right. Any last? Oh, go ahead. Oh, yeah. Come on, you can turn off your mic. There you go.
It's Layla.
Layla, there you go.
Welcome.
Very nice seeing you again. Hi.
So, thank you. I wanted to know if your extinct form can be used for datation to date. If the extinct species of
Yeah.
the Diploneis, we can use them to date the core if we found them elsewhere. Thank you.
This is a very interesting question, but I think it's also a bit tricky because, first of all, as Mark pointed at the very beginning, I mean, we have one drill core that you are really hoping that you will find the species or they preserve in that core. And although with this model, which works relatively well to estimate uncertainties in preservation, meaning to be sure when exactly the species evolved. I mean, the time when it appears in the core, it doesn't mean that it's the timing when it evolved. It just means that it was able to preserve at that very point of time in the core. So that's why this bird death preservation works in order to estimate uncertainties. What is the likelihood for the species emergence? But whether you can use this for dating, I mean, you could, but I don't know how reliable that would be, I have to say. I have to think about that, but I think usually people tend to not use such records or geological events for calibrating something. But it's possible, not that it's not. I just think that maybe you should just leave a little bit of uncertainties or preservation bias age estimates for such an approach.
For the ornamentation that you show for the different Diploneis, is there a better, a more evaluate one? So we can at that moment use it for dating. I mean, you showed us too many different ornamentation in your extinct species from
Yes,
Diploneis,
yes,
for
exactly.
instance.
Yes, yes.
So maybe if you know the function of each information, ornamentation, maybe we can talk about dating.
Maybe I still have to think about that. At the moment, I'm really unsure for such an approach. It's an interesting aspect. I mean, it's an interesting one, indeed, but I have to really think about that. It's very difficult at the moment to say, because I don't think that the function will differ among the species. I think that the whole group will more or less have the same function. Just in some would be maybe they are larger, which is true. Some they are larger, more extensive. But yeah, I really have to think about this. Sorry,
Thank
Lyle.
you very
But it's
much.
a really good question. It's a really good one. Yeah.
Thank you.
Good answer.
Thanks. Thanks, Leila. Well, we reached the top of the hour here. I want to say thank you, Etsy. That was wonderful. And I am so looking forward to seeing the results of this bigger study. I think that question of adaptive radiation, and in particular, what these little structures mean on the diatoms is something that we all just sit and scratch our heads about. And I love that you've come up with a strategy to answer those questions and see what some of these little differences that we see, what they actually impart in terms of adaptive potential for these friends. So thank you so much for joining us.
Thank you, guys. Thank you for inviting me. It was really great to see you all.
Thanks,
Hopefully,
Etsy.
I'll see you sometimes
Wonderful to see
in
you.
one of the meetings, if not earlier.